Hello, everyone. My name is Anantha, and today I'll be talking about active tuberculosis. Tuberculosis is caused by Mycobacterium tuberculosis. The bacteria itself has a waxy cell wall due to the mycolic acid it contains. The cell wall is hardy, which resists weak disinfectants, and so that it can survive on dry surfaces. The bacteria itself is rod shaped. It needs oxygen to survive. Which is why it is mainly located in the pulmonary or near the lungs, or extrapulmonary, like in the blood, where oxygen is easily available. If looked under a stain, it is bright red in color. Um, and the first, I would then like to continue with how it is interacted in the body. So once it is introduced to the body through a cough or droplets from a person with active tuberculosis, the bacteria reaches alveolar macrophage um, in our lungs. And alveolar macrophage are known for eating foreign cells, digesting, and then destroying them. For tuberculosis, they recognize foreign protein on the cell surface and phagocytize them and package them in phagosomes. Then they fuse them with lysosome, which contains hydrolytic enzymes, which break down any biochemical molecule or bacteria. But in tuberculosis, it's tricky since once the bacteria is inside the macrophage, they produce a protein that inhibits the fusion with lysosome and allows the mycobacterium to survive and proliferate, creating a localized infection. And everything I said can be found in the links provided below. So, continuing to healthy physiology, Mycobacterium tuberculosis is inhaled, as I said prior, through a cough or sneeze of a person with active tuberculosis. Um, After 3 weeks of the inhalation, a cell mediated immunity kicks in. So, for primary tuberculosis, which is latent tuberculosis, immune cells surround site of tuberculosis and create a granuloma, which is a wall of bacteria to prevent them from spreading, which leads to death of the tissue in the middle and form caseous necrosis, which is a cheese like necrosis. So a gone focus would be a caseous necrosis in lymph node, which then undergoes fibrosis and calcifies. Um, so due to the calcification, it is seen under X-rays, like in the image on the right. Here we can see how the calcified granulomas are being seen. So, in certain cases of tuberculosis, the bacteria is killed off by immune system or are alive due to being viable but remain dormant. So, in active tuberculosis, it is basically the dormant TB cells break out due to diseases or illnesses that lead to poor immune system. Um, so then the gone focus is cavitated and spreads throughout the body, causing the mycobacterium to be infected. And everything I said could be found on the information down below. So, continuing to epidemiology, the prevalence of tuberculosis is in 2016, World Health Organization (WHO) reported that 10.4 million people were infected. 1.7 million died of the 10.4 million people. Incidence is over 2 billion people are infected with tuberculosis, which is around 30% of the world's population, and 45% of the cases are from Asia, 25 of those cases are from Africa. The regions mainly I looked at were India, Saharan Africa, Micronesia, uh, islands of Southeast Asia. And in those countries or regions, there were about 100 cases per 100,000 people. In China, Europe, Eastern European nations, Central and South America, along with North Africa, there were about 25 to 100 cases per 100,000 people. In the US, Canada, Japan, Western Europe, Australia, they had the lowest rates. Less than 25 cases per 100,000 people. And I concluded that the regions with low income countries were highly affected due to not receiving proper care or treatment on time. So moving on to age, in 2021, 4% of US TB cases were in children that are younger than 15 years of age, about zero point 0.4 cases per 100,000 people. And in 2021, people that were older than 65 years of age had highest incidence rate of tuberculosis, which was about 4 cases per 100,000 people. So, in terms of gender, male contributed more to the tuberculosis cases in the US, which was about 61.3% of 100% people. Male prevalence rate per 100,000 people was about 488 cases, while females contributed to about 231 um, cases out of the 488. And the reason is unknown. It could, in some uh, resources, I saw that the cases in women were less due to the hormonal changes that kept happening more often. Um, but it's not being guaranteed. On the right, we can look at the image that shows the group that I was talking about out of 100,000 people population in the United States. Moving on to signs and symptoms. So, for people with active tuberculosis, the signs and symptoms include coughing for 3 or more weeks, coughing blood or mucus, chest pain or pain while breathing, coughing, sudden weight loss, unintentional sudden weight loss, fever, fatigue, night sweats, chills, loss of appetite. For latent TB, or also known as primary TB, there weren't really any symptoms seen. People do not feel sick, and in that stage, they also cannot spread the tuberculosis. Everything can be found in the links down below. In the image on the left, we can see that fever, night sweat, persistent cough, and just coughing up blood, chest pain, everything that I list out is abbreviated. So moving on to diagnosis, it is so there are about 4 tests, mainly, that are done. So the first one being the tuberculin skin test, or the Mentox test. In this, the an a 1.1 milliliter purified protein derivative is injected intradermally, and then observed from 48 to 72 hours, and then measured using a ruler of the size. So if the size is less than 5 millimeters, then the patient is not at risk, or is at low risk. Usually, less than 5 milliliters is considered a negative for the for tuberculosis. Intermediate risk would be less than 10 milliliters, or that would be when the person is shown to have tuberculosis. And then high risk would be 15 or higher and need urgent care. Um, so then comes the chest x-ray, which comes after the skin test, if the results are positive. And as I said earlier in the slides, that calcified granuloma is seen near the lungs area in an x-ray. Then comes the septum mycobacterial culture. So if the chest x-ray shows positive results, then samples of mucus coughed up is tested. And so this is also tested for drug-resistant strains of tuberculosis. So then comes the blood test, which is a quantiferon TB Gold, or QFT, test that looks for inflammatory cytokines like interferon gamma. So this test aids in detection of Mycobacterium tuberculosis, and it uses the IFNY. Um, then comes the BCG vaccine, which is a single blood draw. BCG stands for Bacilli calmeter guerin, and this is given to infants and small children. BCG vaccine uses Mycobacterium bovis, which is a TB-causing bacteria. So in this vaccine, bacteria is weakened in the vaccine and given to infants to build immunity towards it naturally. The main disadvantages of this vaccine is that the vaccine is of high cost. there is a great amount of technical expertise to um, perform the test. There are high rates of false positive and false negative seen um, in TB skin test for people that have taken BCG vaccines. And on the right, we can see the for the skin test that I was talking about, of how it is measured and how it looks if in all three of the risk stages. So moving on to pathophysiology, the histopathology of tuberculosis is an epithelioid granuloma with that has a central caseous necrosis. Tuberculosis are round nodules with 3 to 4 cellular zone with central caseous necrosis. Intercellular zone contains lymphocyte, macrophages, epithelioid, and giant cells. Outer cellular zone of lymphocyte um, is immature macrophages and just plasma cells. Healing lesions rim of fibrosis is also seen. In caseous necrosis, there is usually a low pH rate, low oxygen tension, and higher fatty acid levels. And all these factors inhibit growth of tubercle bacilli so that the infection is not spread throughout the body. So, caseous necrosis is formed when the tuberculosis bacteria infects tissue and triggers immune response. Granuloma becomes caseous when immune cells die and release enzyme that break down the tissue nearby. Um, Caseous necrosis contains dead immune cells such as macrophages, neutrophils, along with tuberculosis bacteria, and just cellular debris. Typically white or yellow in color, and it has a soft, crumbly kind of texture, just like cheese. We can see that on the image on the right, that there's a cheese like round ball. This is a picture of a lung right in the middle, and that kind of denotes to how a caseous necrosis in the alveolar looks like. So it has many effects. So caseous necrosis has many effects on the body depending on the location, such as it leads to coughing and difficulty breathing if it's in the lungs, or it can cause organ damage if it's in the blood. Um, and everything I said could be found in the links down below. So, getting onto the biochemistry, we see that gluconeogenesis also plays a role. So, what is gluconeogenesis? It's just a metabolic pathway that enables synthesis of glucose from non-carb precursors such as amino acid, lactate, glycerol, and pyruvate. Essential. So the gluconeogenesis is essential pathway for tuberculosis. As I said, it allows the bacterium to have the supply of glucose even when carbohydrate sources are limited. This involves enzymatic reactions that convert non-carb precursors into glucose. So key enzymes involved in pathway include pyruvate carboxylase. So basically, pyruvate enters the bacterial cell and converts pyruvate phosphate by enzyme pyruvate kinase. So pyruvate phosphate is then converted to oxaloacetate by the enzyme pyruvate carboxylate using bicarbonate and ATP as cofactor. Oxaloacetate converted to malate by enzyme malate dehydrogenase, and then malate then enters the TCA cycle and then oxidized to produce energy or ATP. On the image on the right, I've just shown a basic layout of how glycolysis leads to pyruvate, and then pyruvate kind of leads to acetylcholine CoA, and then citrate, citric acid cycle, oxaloacetate, and then through aspartate, and then gluconeogenesis, and so on. So, moving on to the main part, the fate of pyruvate depends on how much acetyl coenzyme A is available, and by the availability of fatty acids in the mitochondria. So, when fatty acids are available, beta oxidation leads to the production of acetyl CoA. Enzyme itself. So then they enter the Krebs cycle and lead to the production of GTP and NADH. When the energy needs the energy needs of cell are met, oxidative phosphorylation slows down. So NADH and NAD over NAD plus ratio increase, and NADH inhibits the citric acid cycle, and acetylcholine accumulates in the mitochondrial matrix. So acetylcholine, CoA here, is used as a positive allosteric effector of pyruvate carboxylase and negative effector of pyruvate kinase. So it inhibits pyruvate dehydrogenase complex both through feedback inhibition and phosphorylation through the activation of specific kinase. So on the image on the right, we can see how the all the blue arrow is just gluconeogenesis, and the red arrows show how pyruvate leads to citric acid cycle. So we're focusing on the gluconeogenesis. So pyruvate um, has a positive allostator effector of pyruvate carboxylase, and that leads to oxaloacetate, and then that is called the gluconeogenesis, and then produces glucose. So then moving on, here on the left I have the structures of NAD plus and NADH, along with pyruvate and how pyruvate kind of transforms into acetylcholine down below. And on the right hand side, I have an image of the GTP structure. So basically, pyruvate carboxylase enzyme just catalyzes conversion of pyruvate um, of oxaloacetate, which is a key factor in the TCA cycle that we just looked at in the previous slide. The function of this enzyme is involved in enpleurotic reaction that replenishes intermediates in TCA cycle. In tuberculosis, TCA cycle is the most important um, precursor for biosynthesis and production of energy. Regulation of pyruvate carboxylate enzyme in TB is believed to be by factors such as availability of pyruvate and the need for energy or biosynthetic precursors. It is important, this enzyme is important due to it allowing bacterium to produce energy and biosynthetic precursors in absence of glucose or carbs. And then enzyme is involved. So the pyruvate carboxylase enzyme is involved in production of mycolic acids, essential component of TB cell wall that we discussed in our health physiology slide. So inhibitors of pyruvate carboxylase have been identified, including drug phenylacetic acid. Okay, so moving on to treatments, wearing N96 mask to filter the bacteria is a good option for people with risk of tuberculosis. Medication for active tuberculosis include isoniazid acid. It's a combination. It's taken with vitamin B6 to prevent neuropathies. Side effects include liver injury, nausea, rash, numbness, tingling in extremities, and headache. Rifamycin is a medication followed up with liver function test done due to it having a history of causing hepatotoxic. Since it is hepatotoxic, um, the side effects include jaundice, discoloration of urine, secretion, along with discoloration of secretions of the body, um, arthralgias, and fever. Um, Ethimobutyl is used to. It is highly avoided in children that we cannot measure the visual test or vision on, since this medicine is known to cause optic neuritis. The side effects include blurred or decreased vision, blindness, liver injury, headache, and nausea. And then the fourth type of drug used is the um, pyrazim, pyrazinamide. This is given. This is usually a medicine that causes nausea, painful or swollen joints, or liver injury. And patients are given like periodic liver function tests, since it can cause liver problems or injury. Chest X-ray, serum uric acid, and sputum cultures done at 2 to 3 months, and the completion of the treatment. On the right, I have shown an image of just how if everything falls in place, the risk can be lowered. Starting with the right patients, it just means that the low people that have low risk rate, moderate or high, and then how that moves to the type of diagnosis and then to the right treatment. So usually, to look out on this image is to see how people that diagnosed with low risk have a treatment duration of 4 months. People diagnosed for moderate risk have a duration rate of 6 months, and people diagnosed with high risk have treatment duration for 9 months. Okay, so just an interactive assessment. Just draw like the structure of the NADH and just NADH, or just point out the difference of what you remember, and then here's the answer. So thank you so much.